seminar afternoon. So we are going to start. If we can have some silence, thank you. So before we start with the seminar, there is a change in the schedule for next week. So on Monday, you will have class with Mariana and Daniel at 9 and with me at 11, okay? So there's going to be a switch. Again, my class, we have the exam in the afternoon, so we are not going to do anything new. It's in case you want to ask me something. I don't recommend you to wait until Monday to ask, but it's up to you. You are all adults and you know what to do. So we have today's speaker is going to be Flavia. You all know Flavia as a person, very good one. As a scientist, Flavia is both a biologist and a mathematician. She got both degrees from the University of Campinas, from Unicampi. She also got a master's in ecology in Unicampi and a PhD in ecology from the University of São Paulo. The master's and the PhD under the supervision of Paulo Guimarães, who is an ecologist here in Brazil. And in the PhD, she did some short-term visit in Princeton University in Simon's Levine group. And then since 2015, she's a postdoc in Unicampi in Marcus de Aguiar group. And in that time, she also did a visiting fellowship in Princeton in Simon's Levine group when it's where we met in the second visit. And since this year, she's also a independent researcher here at ICTP-SAIFR. And we are very happy to have her here with us. And her work is very broad. She's worked on a very large variety of topics, always in the interface between applied mathematics, ecology, and evolutionary biology, in very diverse topics related to the evolution of cooperative interactions in ecological networks, looking at the effect of space in the emergence of diversity in ecological communities, and many other topics that she will give us an overview today. Besides research, she is also a very active teacher and a mentor here in Brazil in different programs. So she regularly teaches both at the University of São Paulo and the University of Campinas in the ecology program. And she mentors a lot of students in both programs as well, both masters, undergrad students, and PhD students. So with that, thank you very much, Flavia, for being with us every day. Everything is yours.
Okay, I will remove my mask. You remain with yours. And now you see my face. That's very rare, right? Is it too loud? Okay. So thank you, Ricardo, for this very nice introduction. Don't sleep, guys, please. Thank you very much for this very kind introduction. And in the next hour, I hope only 1 hour, I'll be talking about my career. And as you see, I put a title that I was telling the guys that I hate. Very, very broad. It seems I'm a star in ecology and evolution. That's not the point. It's just hard to generalize some things. So I'll be talking about topics in ecology and evolution that were part of my career and are part of my career so far. And I do believe that collaborative science is what makes science nice, is what makes science great. And I've been collaborating with many people. I've been supported by good agencies and by good universities. And fortunately, the list of colleagues and labs that I've been passing through my life, it's bigger than this. But I chose to put here the names of the works that are related to the things that I'm presenting now. But I want to let you know that collaboration is great and what makes words wider. And I would like to thank all these people. And of course, thank my fundings that I had so far. And it's a very complicated moment in Brazil, and I was very lucky to be funded until now. That was really hard. And so since my first bachelor, that was in biology, I'm fascinated by interactions, by ecological interactions mainly. And I've been working since the beginning of my career with mutualist interactions. So mutualist interactions are those interactions between individuals of different species. So you have interactions, like seed dispersal by birds or other animals. You have pollination, that is the act that some animal does the transfer of pollen to another plant. You have, for instance, oh my God, I never find the button here. Some animals that clean the tissue, the dead tissue of other animals in coral reefs. And you have mutualist interactions as well in Müllerian mimicry rings. They give it the same signal to their enemies. So all these kinds of interactions are interactions where both parts get some benefit, or both parts are actually exploring the other part. As time passed, I got very interested as well in understanding cooperation. And cooperation, and it's an interesting kind of interaction, and how it evolves and it is maintained in nature is something that is very studied in many, many fields. And not only in biology. And you can find the cooperation evolution in different groups. You can find cooperation evolution, for instance, in ants, where the work ants, they don't reproduce, and they help the queen to maintain their nest and maintain their offsprings. You have these, how is it, surrogates? Surrogates. Oh my God, I don't know animals in English. Very theoretical, right? Sorry. So the surrogates, they, some of these individuals of a burrow, they stay outside of the burrow and they make an alert call to their co-specifics when they see some danger approaching. So they are like in danger themselves, and they are making an alert for their friends. Friends? No, co-specific, sorry. They're not, I don't think they call their friends. You have also hematophagous bats that some of them throw up some food in the mouth of other animals that couldn't find food during the night. So they help each other, like throwing up food in others animals' mouth. And you have also like primates that have like a grooming behavior where they remove, for instance, ectoparasites from their co-specifics. So this kind of interactions became very interesting I became very interested during my career, just because it's how these things evolve, how they are maintained. So these questions came to my mind. And so I wanted to let you know how I started, how my passion started, and how it percolates in my career. So I will be talking about my research, past, present, and future. And future, I'll be presenting some of my ideas and some things that I'm working now. So in the ecology topic, I'll be talking about ecological networks and rate-induced transitions. In the evolutionary part, I'll be talking about game theory and diversification. And I'm not talking, unfortunately, about epidemiology because there are too many topics already. But if you want to know about it, we can talk later. In the epidemiology topic, I worked mainly with the people from Observatório Covid-19 BR, analyzing data from Brazil and proposing new ways to do things and measuring how many deaths, how many people were saved because of vaccination, how we could deliver a better way the vaccines, how was the best strategies that we could take. Many of them were not taken, as you know, but we have shown this. And so in the ecological networks, I worked mainly with interactions between different species. But complex networks, oh, there you have everything, okay. Complex networks mainly are the way that we use to represent interactions between many different entities. Entities here are represented by the nodes, so these little balls, and the interactions are represented by the links. So these nodes can be, for instance, genes, they can be, for instance, individuals or species. And as you like represent things as networks, you can detect the patterns of interactions that they establish between them, among them. And you can make questions about the process underlying these interactions. What are the processes that are making, formatting this kind of networks? And because I'll be talking about ecological networks and I need to talk about some terms many times, I'm defining them for you, okay? So basically, we measure things like the structure of the topology of the network. And usually for ecological networks like mutualist interaction networks, we use this bipartite format where you have, for instance, plants in one side, species of plants in one side, a species of animals in the other side, for instance, for a pollination network, and you link them if they interact. You can have weights and everything, but I'm simplifying here. And there is this pattern of interaction that I represented here that is the nestiness. And the nestiness is just like how we call something like this, where those that have few interactions have a subset of interactions of those that have many interactions. This is what defines nestiness. You can also have a modular network, or you can measure modularity in networks. And so the modules, the communities, or the groups in these networks are the set of nodes that have more interactions between themselves than the interactions with the other, with the nodes of other modules. So I represented somehow here different colors for the different modules, but this is not a very modular network, to be fair. You see here that many of these nodes have only one interaction, right? So it's kind of, but not really, but it's just to represent how we can measure things. So these are like the main things that I'm talking about. I also talk about other metrics and I'll define them. But just to let you know, because of the facility that I had in the beginning of my career with programming and with algorithms, optimization algorithms, I proposed a software called Modular before R was a thing. So it's a software, a real software you can run in your computer just to measure modularity in ecological networks. And I did this because many of the people that came to me asking questions were questions about how can I do this, how can I measure, how can I find modules in my networks. So I did many collaborations because of this. But then I realized that it would be very useful to produce something that people could use. And so I did this, and we proposed it as well, new algorithms to find communities in these in networks. And many people don't know that. I think people that use the program don't know that, but it's also a novelty, like how we propose how to find modules in networks. Just to define two other metrics that I need to tell you about. Degree is just the number of links each node has. For instance, this guy here has a degree equals to 7. This guy here has a degree equal to 1. It's just the number of links it has. And the centrality, and here I'm not defining any metric of centrality, but I give you the idea that what it is. So the centrality is one node. For instance, this guy here is linking different paths. So you can, if you go from this node to this node, you pass through this central node. So the centrality is this kind of idea. And the first work I'm going to present, and I'm going to present works that I love a lot. That's why I chose them. And it's also a good advertisement as well. But in this work, we wanted to understand how individuals of sea otters, they use their resources. And resources here is what they eat, right? So they eat, I don't know if I can say the words in English, but these animals here, okay? So they eat different animals, they have different resources, and we wanted to understand what were the process that were shaping the resource use that they shown to us. So Many of them use different sea otters. They consume their food items in different proportions. For instance, they can eat some item more frequently than the other items, and the item can be different depending on the individual. They have different preferences, for instance. And this is what we wanted to understand better. So we defined scenarios and we tried to understand what would be the patterns that we could observe in the interactions of these individuals with their resources. And what we proposed in the first scenario, the shared preference model, is that individuals usually consume the same resource. Different individuals here are the circles, and diamond is the resource. So different individuals can use the same resource if there is no competition for that resource. And when the competition increases, they start to use other resources in such way that they form this kind of nested network, right? So they complement the resources that they are going to use, increasing the nestedness of the network. In another scenario that we called competitive refuge model, the individuals consume the same resource in a low competitive scenario, and when they increase, the competition is increased, they start to consume different kinds of resources. They escape from their co-specifics by using different kinds of resources. And in a third scenario, we said, oh, maybe they already prefer different kinds of resources. So the network is initially, or in a low competitive scenario, it's very modular, and the modularity of this network should decrease because they will establish new links, and a very modular network will become less modular. Of course, I know this is not well-defined modular network. This is just a graph to represent this kind of idea. This is not a not connects network. And what we found, and here I'm going to present these three plots, but I'm going to represent here the diet fraction remaining. So when I have 1, it means that we are considering the whole diet of the individual. And when we go from 1 to 0, we are going from the whole diet to the most preferred items that they have in their diet. And I'm representing modularity here and nestedness here. And modularity will be the dark, and the nestedness will be the diamond and gray here. So what we found is that when we consider the whole diet, so when we consider the diet fraction remaining equals to 1, the networks are all nested in a low density and a high density. So this is very, how can I say that? This is what is predicted by the shared preferences model that we defined earlier. And if you analyze, as you go from the diet remaining equal to 1 to a more preferential diet, we see that we increase the modularity of these networks in a high density. And this is what is predicted by the competitive refuge model. So here we use tools from the complex or from network theory to understand better how these others are consuming their resources. So we understand the processes behind the patterns that were observed. And just to represent it better, we see that they consume resources in a nested way. So the items consumed by this individual that has few items that it consumes are a subset of the items consumed by the others. And if we look at the fraction that they consume these items, what is happening is that they actually prefer different items in their diets. As I told you before, I just need to see my next slide. Okay. As I told you before, I'm very fascinated by mutualist interactions. And this is another work where we work with the mutualist interactions, and the kind of interaction that we work at here is the interaction between seed dispersals and plants. And this is a network from Pantanal. So Pantanal is the largest wetland in the world and is in Brazil, located in Brazil. It's drying, unfortunately, but it's large, it's still large. And I had the chance to work with these people that really go to the field. I usually don't go to the field. I usually don't observe this kind of interactions. So to make my work to have some biological meaning, I have to interact with these people that know the system very well. So I had the chance to work with these people in the previous work as well, but this one was very, very nice. These are Brazilian people. And as you can see, the network, the seed dispersal network in Pantanal is very modular. And this is the first result that was different for us. So if we compare the seed dispersal from the Pantanal with the seed dispersal networks that we have in the Atlantic forest, that is the biome that we have here in São Paulo, for instance, the one that we find in the Atlantic forest is more nested. And we found in Pantanal one more modular. So this was very interesting for us. And we wanted to understand what was splitting a species in different modules. So just to explain to you how we found these modules, we didn't force them to different groups. We use an algorithm that doesn't know the name of the species, doesn't know anything, that looks only to the interactions between these nodes and tell us, oh, these guys here form one of the modules. These guys here form another module. These guys here form another module. So the algorithm is not looking at the processes that are shaping this thing, right? So we wanted to understand why we could detect some very interesting modules. So we could detect this module that is mainly with birds. This module is with large birds. This one is with mammals. This one with small mammals. And this one with fish. Yes, fish can disperse fruit. That's amazing, right? And because we saw this relationship, we saw that the animals were mainly divided in these groups. We tried to understand if there was any phylogenetic signal. When I say phylogenetic signal, it's a metric, but it just means that I'm looking for an information in the shared evolutionary history that can explain this. Do they have in their genes for a very long time that makes them to be like this? So what we found is that the phylogenetic signal is very strong in the animals group and mainly in the birds group. So the modules that they are is basically defined by the phylogenetic signal. And if we go to the plant side, we don't find that. The colors here is spread, right? And this was okay, so maybe the animals define, the phylogenetic signal of the animals define. But then we decided to understand how the size of these animals could explain these networks. And if we put the size of the animals in the x-axis and the size of the fruits, the fruit diameter in the y-axis, what we see is that the modules form this very nice relationship. So we could conclude by this work that the shared evolutionary history, together with this convergence of traits, can explain the format of the ecological the dispersal networks of the Pantanal. So this was another work where we could relate pattern and process. And what I find very interesting when you're working with different tools and when you have the chance to interact with different people like you here, you have the chance to know things that you never heard about. And this was what happened with the friendship paradox, that I don't know if it's a paradox, by the way, but it's called the friendship paradox, and it comes from the social sciences. So people in social sciences say that on average, the friends of randomly selected people have more social interactions and are more central in the network than the initial set of people. So if I pick random of you here, and then you point your best friends, your best friends will have more links, and your best friends will be more central than you are, the initial group. Nice, right? That's super cool. And so we decided to use this tool in gas, Mutualist Networks, and here we worked with Pollination Network. And let me see, I don't remember what is the diamond. I didn't tell what it is. Oh, the diamonds represent the pollinators and the circles represent the plants. So we get a random one, a random guy, for instance, a random pollinator, and we ask them, which are your best friends? And it indicates us. I mean, we don't ask, we just look at the network, right? And then we have the set of friends. So I'm going to call this random, and I'm going to call this partners, okay? And we tested this in an ecological network, and when we look at the results, we see that it's true. The degree or the number of interactions of the random group is smaller than the degree of the partners. And the centrality of these nodes in the random group is also smaller than the centrality of those in the partners group. And this happens for both plants and animals. And then we decide to understand, oh, they are central, they have a higher degree, but are they really important? And to understand if they were really important, we simulated extinctions. And we simulated extinctions by just removing that node of the network and counting the secondary extinctions that happened in this situation. So the proportion of extinctions here means that if I remove the node or a node of that group, many of others will also have no interactions. And we observed that at least for the plants, it's true, these plants are very important. If we remove them, they are consistently causing more secondary extinctions than in the random group. Not very true for the animals, and these here are different networks. And so in this study, we proposed to use the friendship paradox as a tool to understand if a species is central, if it's a keystone species maybe, in ecological networks, and especially in those that you don't know the network at all. You go to the field, you select some random species, you look those interactions, the interactions of those initially selected species, and then you likely have a group of other species that are central and have a big importance in that network. So this can be used for management or for understanding better your system and also for conservation purposes. And more recently, I started to work on understanding how ecological networks can shift to a different regime. And I think you heard about tipping points here in this course, right? That's good. I'm afraid, but that's good. And we basically, instead of simulating the extinctions that we did in the previous work, here we are really simulating the dynamics, but we still have a network that defines the interactions. And let me remember what I'm supposed to say here. So we live in a changing world, right? We live in a changing world that is not only changing, but it's changing fast. And we understand that maybe changing fast is not the same thing of changing, right? If you change fast, if you change slow, it can be different for the species. The species, if they have time to adapt, maybe it's not a big deal. But if they don't, it can be a big deal. And we decided to study rate-induced transitions in ecological networks. And to work here, we worked with the format-only theoretical networks. And we try to understand if they shift to a different regime. So you have usually the bifurcation-induced tipping point. So you have a control parameter here. And if you change, the system state can go from one state to another state, from a stable state to another stable state, or from a bad scenario to from a good scenario to a bad scenario. And good and bad here are defined depending what you want, what you study. And if you go from here to here, if you increase the control parameter, you change from this to this. And if you go backwards, maybe you have to go to a lower value of control parameter than this one that defines the first shift. And this is called hysteresis. And you also have other kinds of tipping points. You can, by noise, for instance, provoking noise here in the state you are, you can cross and go to the other state. And the one that we're studying and I'm presenting here is hard to represent in a plot, but the idea is that the control parameter is actually moving. It's going fast. It's not actually this ball that is moving. What is moving is actually this thing here. And so we did this for pollination networks. We worked with the nested structure, so we worked only with networks that are theoretical, but based on the same structure that we observe in nature. And we found the bifurcation parameter that was, we found the parameters that was making this system going to a different state if we were going forward and if we were going backward. This is just a summary of what we have here. And then what we decided to do was to actually change the rate that we were moving our parameter. So here we have how the parameter is changing. And I'm going to show you here what we did. So this full collapse here is the one that is predicted by this value here, okay? So we have this threshold here. And what we did was going to different thresholds, one high, one medium, and one low, slowly or fast, okay? So when we go fast, we are here. When we go slowly, we are here. And we are going to very close to the full collapse predicted by the previous model, and an intermediary value and a low value. So the orange one is the close to the full collapse. So if we were here, we would not collapse. We would not move to the different regime or to species extinctions. But here we showed that if we go fast or fast enough, you move to this different regime. And not only that, what we show is that if these networks or if these species have the capacity to rewire or to rewire, no, to rewind, my God, I cannot say that. Redistribute the weight of their connections. What happens is that it's even worse. We can have some collapse or an earlier collapse in scenarios that were not happening in the previous situation. So this is why we think a new theory that is almost set, but we need to study it better. The world is changing fast. The way species adapt may have a play in this situation. And we want to understand that better. That's why we were developing this kind of work. And on the future works in this topic, I'm working this, in these Tri-Trophic Interactions. And here we have, for instance, crops. We have the pests that use these crops, that eat these crops. and we have the natural enemies of these crops that are in these plants as well. So we have plants that we plant, for instance, apples, lettuce, this kind of thing. And we have the animals that eat the leaves of these plants. And we have the natural animals that prey upon these animals or do something bad with these animals, these pests, and also use the same plant. So we are constructing these Tri-Trophic interactions based on data that are available online for the crops that we have that have agricultural interests for Brazil, especially, and constructing this network to identify if we can propose consortium-mediated biological control, which means can we put two plants together that share a common natural enemy of some pests. We want to propose better ways to use natural resources. And of course, we understand this data available online, they're not enough, they're not good enough because they don't have, for instance, data, oh, there are some animals that live here in São Paulo and consume sugarcane. I don't know if sugarcane is consumed by anything, but let's say it is. But this animal cannot survive, for instance, in the northeast of Brazil, but we can plant sugarcane in the northeast of Brazil. So is there any way to improve that? And this is a project of citizen science that I want to do in the future and collaborate in the future with people that are actually planting these crops, that are actually in the field and can give us information of the kind of pests and natural enemies they have in given spatial geographic distribution. So now passing to the more evolutionary part of my talk. I don't want that it looks like that evolution and ecology are separated. They are not. But I had to split like this because it was easier, guys. So as I told you in the beginning, I'm very interested in understanding mutualist interactions and also cooperative interactions. And unfortunately, this is not a work of cooperative evolution or cooperation evolution. It's just about, it's not just, it's a very beautiful work of evolution of different strategies in these lizards called Utah lizards. Many of you may have heard about it. So these Utah lizards, the males of these Utah lizards, they have a different color in their throat. And some of them have yellow, some of them are orange, some of them are blue. And, and the behavior of these males and how they behave to mate with the females is related with the color of their throats. And not because the color attracts more or less the females, it's just about the behavior that they have makes the color change in their throat. So here they have different colors. And so we call these different strategies, right? And because of that, I decided to use, I know, Cinerva and collaborators already proposed that. We are using their work to construct ours from theirs. So they proposed that the way that the males fight against each other, it's not really a fight, can be explained by a payoff matrix like this. And what happens is that the yellow one, I need to double check, but the yellow one, no, let me start with the blue. The blue one is the kind of male that protects their territory very well. And because they have a small territory, because they have to protect this territory, they keep very few females in their territory. And the orange one is that guy that has like a very broad area, but cannot protect the females very well, but has many females. And the other guy, the yellow one, is the sneaker one, is the one that steals females from the males. So it doesn't maintain any territory, but it can enter, for instance, in the orange territory because it's not very well guarded, and steal the females from there. And this can be explained by this payoff matrix. So here we can say that the blue one that protects few females can win against sorry, the orange one can win against the blue one because it has more females in its territory, but when it's naked, when this guy here enters in its territory, it gets a lower payoff. And you can represent the same thing in the other lines, and what happens is kind of a Jacques and Paul kind of game, and like rock, paper, scissors. And what we wanted to do is to include this strategies game that was already defined by Synerva collaborators with the genetic system that defines the colors of these males in the same model. And here is also work done by Synerva group. They have a very interesting broad work in the game theoretical part and the genetic part of these lizards. But these things were, as far as we know, these things were never matched in the same model. So we decided to join these things. So what we are doing, we are incorporating the rock, paper, scissors and the genetic structure. And just to explain to you, the genetic structure is like this. So they are diploid individuals. Always they have a no, they are orange. If they are yellow, at least with another yellow with the blue allele, they are yellow. And the blue one is this homozygote. Oh my God, that's hard to say as well. And so we have this kind of relationship between the alleles, how they predominate. So I'm going to present a set of results. Just to let you know, I'm presenting here the same payoff matrix that I presented earlier, the rock, paper, scissors, but here I'm changing the values of A and the values of S, okay? Just that. And what we observed is that the value of A is here, the value of S is here, and what we observe is that these three strategies, rock, paper, scissors, orange, blue, and yellow, can coexist in this area of combination of A and S. And here, they cannot coexist. Just one will remain. And we have the same for the genotypic one. So we studied, and we studied where this line was located. And what we saw is that when we combine the rock, paper, scissors with the genetic system, the stable area increases. And just to show you in another way, so here I'm overlapping those previous plots, and we are going to see the results for the green and for the orange points. So here, they're both independent of the model we are using, just phenotypic or phenotypic plus genotypic. We see that the coexistence is stable for both scenarios. And the orange point here is the situation where it was unstable for the original model, the phenotypic one, but it's stable for the genotypic plus phenotypic one because it's here. So this is what we found. And we did it, we moved A and S to different positions in this payoff matrix, and also incorporating the genotypic information, and we could define the different lines where you cross from unstable to unstable equilibrium. Let me see what comes next. So the conclusion about this work is that when you include more biological information, it can be more complex, it can be more complicated to analyze, but you gain some kind of information that you cannot have if you do not include that kind of the genetic part, right? So this was, the game theory is a very well established theory. It's easy to analyze, but we need to understand that biological systems also have other components that can be important, right? And now passing to, I think from now to the end, I'll be talking about this kind of models that are the diversification models. So species diversity, how they emerge, how they are stable, how you have the different species, how they form, it's one of the big questions in biology, right? People want to understand why we have more species in the tropics than we have in the non-tropical region. Oh my God, I forgot the word. And how they emerge, how these species are formed, how they go extinct. These things seem very interesting to many people, and for me as well. And I wanted to understand, sorry, I'm looking at the wrong slide. And I wanted to understand not only how these species are related to each other, for instance, what is the evolutionary patterns by, for instance, a phylogenetic tree that give us the information of how they are related, but also what are the shaping processes that are making these species to emerge. So I wanted to understand, for instance, if how they use the geography, how they use the space can be an explanation for how they are organized in these phylogenetic trees. So I wanted to actually join this kind of information, look to the macroevolutionary patterns, understand what are the microevolutionary forces, and build the gap that is between these two things. So that's how it started. And mainly when we talk about diversification, the geographic configuration plays an important role. I think many of you, independent of the field you are from, you heard about in the high school that if you put a barrier between individuals, these individuals accumulate differences, and then when they meet again, they cannot reproduce anymore. So they form, for instance, different species. Defining species is really hard, but this is one of the ways that we explain how species emerge. And this is called allopatric speciation. And this explains, for instance, how some primates emerged in Congo, how they speciate in Congo, because it was a Congo River that separated a population of these primates and formed, I cannot say the names, chimpanzee, bonobo. Okay, so formed these different species. And there's another way of species to form that is called sympathetic speciation. So in the sympathetic speciation, you don't have any barrier, you don't have anything physical that tells you that these individuals are using different environments or something. It's just because they somehow accumulate differences and are divided in different groups of individuals that we call species. And the sympathetic speciation is something that people don't accept that well in biology, but we try to understand. Even if it's not that well accepted, we try to understand. And many of the ingredients that are used to explain how species can appear in a sympathetic way is using assortative mating, which is someone prefers to mate with someone else because of a given trait, for instance. By gene linkage, if there is some gene that is linked to another gene and forces a differentiation like this, or by competition. If they are using different resources, they split because they're using different resources because they don't want to compete. One of the most well-known works in sympathetic speciation in biology was done by Dickman and Unveiling in 1999. And this is the main reference in this kind of work, and it's very theoretical. Nowadays, there are many people finding species like ants in the Amazon. No, it's not ants, it's something, tapir, antes, that I don't remember the word in English. Tapir, tapir, okay. It seems they evolved in a sympathetic way, and that's interesting. And what I decided to work was with a different model, a more simple one with less ingredients, at least with no competition, with no assortative mating, with no any other ingredient that we could call to explain the sympathetic speciation. And this model was initially proposed by Derrida Hicks and the Hida and Higgs and the Hida. They are two physicists. These works were mainly published in physics journals, and so didn't come to attention of many biologists. But this is a model, and it's very interesting. We decided to make it well known in biology. And the Hida-Higgs model, they use a different kind of model that I'm presenting here, but I'm telling where the differences are. And they analyze it very well in well-mixed population, well-mixed genes spread, but we do an individual-based model for this analysis. And why we do an individual-based model? For two things. First, because the Hida-Higgs, they use an infinite genome, and we use a finite one. Why? Because genomes are finite. Just because of that. But it's very interesting to understand things more broadly, right? What would happen if the genome was very, 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 very large? You would be a salamander, but, no, I'm kidding. Salamanders, by the way, they have a huge genome. But, and an individual-based model was also chosen because we were using a spatial model. And why were you using a spatial model? Because of the reasons that I explained before. This spatial model, the spatial configuration is something important in biology to explain how species emerge. And we wanted to confront or understand how this could be playing a role here. And so we work with this kind of model where we have a genome. Individuals are defined by a genome, a very simple one. We call biallelic. You have only zeros and ones. So each position here, each loci, each gene, we don't know how to define that, but it can be 0 or 1. And it's a very basic model. I can explain in a few slides, and I hope you understand. But it's simple, and it's nice how it produces something that we can understand. So we have a focal individual that has this genome, and we have a sexual partner that have this genome. They have zeros and ones. And they can reproduce if they are compatible. And how we define they are compatible? We give a distance g, for instance, 4. And if the differences that they have between each other, the focal and the sexual partner, is smaller than g, they can mate. If it's greater, then cannot. It's very generic. I'm not defining that they need to be the most similar ones. I'm not defining they need to be, the reproduction is likely to happen depending on the distance. I'm not defining that. I'm defining that I have a distance g. Below that, anyone can reproduce it or anyone else. And if they can reproduce, they form an offspring here that will form the next generation. And each loci here will be, with 50% will come from the focal parent, with 50% will come from the sexual partner of the focal parent. And so here, we don't know from where it came from, but we know that here it came from the focal one, and so on. And we also have a mutation rate. So here it was supposed to be a 0 because in these positions both parents have a 0, but then it mutates and changes to 1. So we have very few ingredients. And then we put these individuals in the space. And the space is the one that you were learning with Ricardo. It's a lattice with size L. And then we we call periodic conditions. So we join this part to here. We join this part to here, right? You heard about that. And if you imagine this kind of figure is a kind of donut, yeah? And what we do is that for each individual, we give the chance to reproduce. And when it has the chance to reproduce, it looks for a mating partner in this mating range area. It can be a small mating area or a large one, right? It can be any we define. And we need to define a species. I told you that defining species in biology is very complicated. We have, I don't know how many definitions for a species concepts in any flavor you want. But here we're using a very simple one. So I worked with networks, right, for a very long time. So I said, oh, good, I can see networks everywhere, including these models. So if we link individuals, if we consider that one individual can reproduce with the other, their differences below G, we establish the link. If they cannot reproduce, we don't establish the link. So we can form a network like this. As time passes, individuals reproduce, mutation happens. These individuals can split in different subgraphs or different components of these networks. And this is what we call a species. So this is a different species of this. These three are different species. So we are defining species here based on the genetic flow individuals can have. It's not that they really have, but they can have. So as I told you, in this very simple model, we start with a homogeneous population. Everyone is equal to everyone else. They reproduce, they mutate, and they form species that are defined here by different colors. And analyzing the different mating ranges that we have, we could understand better how these spaces formatting or shaping the species formation here. So we work at, I will focus mainly on this part here, okay? So if we start with a large genome and a very large mating area, in such way that I can call it sympathetic speciation, we see that we form less species, but very fast. So there is a very fast radiation process. Species is form very fast, and then they maintain a given number that is lower than when we have a smaller mating area. And when we have a smaller mating area, because we cannot call allopatric because we don't have any barrier, we call petapatric, okay? So we analyzed it, and then we understood first this, right? We form less species in sympathetic than in allopatric, and the radiation is very important, it's very fast. And if the genome has also a play here, if the genome is smaller, it takes a longer time to form species and equilibrate in a given equilibrium, and that's it. And we wanted to understand other things. For instance, how these species are related to each other. We had all the information. Remember, we had the individuals, and the individuals were splitting species. So we were following every event that was happening. So we could annotate and record all the speciations and extinctions events in this process. So we could follow everything that was happening and put in some archive, some output file, what were this information for speciation and extinction. We could also use some other different methods, and this is the most recent common ancestor method, to detect how these species are related to each other. And this is just because the previous method is very expensive computationally. So we wanted a better method to do more simulations. And so we had everything we wanted. And we decided to analyze those situations that I showed previously regarding their balance, the balance of the phylogenetic trees that were formed, and also the branch length of the species that were formed in these scenarios. And so we got those information, right? We recorded everything, and what we observed first is that when we have this kind of the sympathetic kind of speciation, what we observe is that the phylogenetic trees, in the end of the radiation, they are more TP and very balanced. On the other hand, when they are more petapatric, they are more steamy and still balanced. I'm not talking about this too, but something I wanted to, and sorry. And I want to let you know that we compared, and these dots here, these squares and the triangles, are phylogenetic trees found in the literature that can be called adaptive or non-adaptive. When we say adaptive, there was a process like natural selection or some competition or something else that helped to form that, or was one of the ingredients to form those species. And we also have non-adaptive scenarios where they cannot detect anything that was like the main ingredient to form those species. And we also found in the literature the genetic flow, if it was low, if it was high during the speciation. And when I say it's high, I'm closer to the sympathetic. When I say low, I'm closer to the parapatric. So we related this, but I'm not discussing because I already passed my time and I have a lot of things to say. And just to let you know why the next work comes. So we realized that for large nomes, when we decrease the radius, the mating range that we are analyzing, we call the acceleration of the speciation process decrease it. So we went from more tippy to more steamy network. So the branch lengths were affected. So if we go to a sympathetic to a parapatric scenario, we slowed down the speciation process. And we said, oh, great. Now we can analyze another spatial configuration. We can say, oh, what about if we have barriers, like here? If we have 3 barriers, 2 barriers, 1 barrier, what does it happen? So what we were expecting, based on the previous model and based on the previous work, is that decreasing the radius, we were actually making the environment more structured, right? We were going to from a sympathetic to a parapatric, to small balls. And now we are including a more structure to this spatial configuration, so I was expecting that I was going to slow down even more the speciation process. And it didn't happen. Sounds obvious, but it didn't happen. What we observe is that it doesn't affect a lot, depending on the number of the demes or number of islands we included. It doesn't depend on anything. So the acceleration is about the same. And I said, oh, boring. And what happens if we analyze the balance of these phylogenetic trees? And then we got surprised. So, and here I'm putting the number of species in the x-axis because the balancing of phylogenetic trees depends on the number of species in the phylogenetic tree, okay? So what we observe is that if we go from a configuration with only 1 island to a configuration with 4 islands for the same number of species, what we see is that the balance of in the 1 island is the network is more unbalanced. The phylogenetic tree is more unbalanced than it would be in a 4 demes configuration. So these islands were playing a role in the balancing of these phylogenetic trees. And then we decided to analyze these phylogenetic trees that we know from nature, and that was very interesting. So for this species here, this is the how is called a silver alliance, some plants from the Hawaii. These are the Tretagnata spiders, and these are lichens, Ticta lichens. We could predict the number of the demes or islands that they evolved based on the balance of these phylogenetic trees and based on the number of species that we had in these phylogenetic trees. I cannot say that it was this that happened, but that's a very strong signal, right? And however, and when we look at the Darwin finch, we didn't find that. And the Darwin finches are like the most cute system in evolutionary studies, but we could not predict the number of islands they evolved under based on the number of species and based on the number of, on the balance of their phylogenetic tree. And why? Maybe because they fly. that's the explanation we found. And this was the work that we developed. And still currently, I'm working on the constraints of sympathetic speciation and trying to understand how sympathetic speciation is really a thing. And this came from a very practical problem. We wanted to simulate 4 small environments to be one big environment. Why? Because it's cheaper computationally. So we wanted to understand if we sum these 4 things here, does it mean the same thing of one large thing like this? We wanted to understand that. And it seems that when we have a parapatric situation, when the mating radius is small, it's true. We can sum 4 small simulations to be one. And the number of species it's maintained, we have about the same number of species. And the species size, and when I say species size, is the number of individuals in each species, the frequency here. And we observed that this distribution is about the same. But when we use a larger mating area, so we are going to the sympathetic speciation, we cannot even sum. The distributions are very different. This is only one example, but we cannot sum. If I have a small environment and my lattice has a size of 50, and I want to do 4 simulations of 50 instead of 1 simulation of 100, if the radius is close to 50, it's close to the whole environment, it cannot happen. And what we found was this relationship here, and we are still studying this, but just to make a summary of the results, we defined the line here. We can find this line here. It's where the simulations can be summed. So you can sum simulations of different lattice sizes and different mating ranges if the points fall in this line. So we were trying to understand, is sympathetic really sympathetic? Can it happen all the time? So we were trying to understand what is the scalability of the speciation in these scenarios. And more recently, it was just published, we started to work the reticulate evolution. What is reticulate evolution? So reticulate evolution is when we consider that a species actually don't see each other as, individuals don't see each other as different individuals, right? They don't know if they're individuals of the same species. Like, imagine 2 beetles. Do they know they're individuals of the same species? I don't know. I don't think they know very well. And that's why hybridizations happen, right? Individuals mate with other individuals that were already considered different species. And then we have, if we had 2 different species already separated here, if they started to reproduce again, the gene flow is reestablished, right? And then we have a hybridization. And you can also have extinctions. And when you consider this kind of things in a phylogenetic tree, so consider here is a branch of phylogenetic tree. If we have an extinction, this branch ends. If we have fusion or reversal that are only 2 names for hybridization, what happens is that they split and they join again in the phylogenetic tree. So usually we see phylogenetic tree as binary trees, right? The very typical ones that we see, even in like mathematics, we see binary trees in many, many times. But here we are considering that we have loops in these phylogenetic trees. And this is a very simple work, but it was very important to our understanding of the model. It was done in this model, it's very theoretical. We don't compare it with anything in nature, but we need to define what happens in this model and what happens in this very simple and neutral model to understand what happens when we complicate it. So we decided to study in this very, very simple model. Oops. And what we see is that extinctions happen all the time, and it has a given rate that is happening, that is constant. And this is just the cumulative, and here is the number of events per generation. And don't worry about that. But we see here that the genome size is not important for the number of extinctions that happen. However, for hybridization, the size of the genome that we are considering is important, or the amount of genetic information that we are using to define species is important. And more than that, fusions are more common than reversals. And reversals is just something that happen after two speciation processes. And until this last work, we were doing everything in this very, very neutral model. We don't consider that species compete. We don't consider there is a very assertive mating as defined in nature. We don't consider anything. And then because we were working with cooperation evolution, and we were trying to understand cooperation evolution in other scenarios, we decided we wanted to understand also the cooperation evolution in a scenario where individuals can split in different species. That's weird. That's not common in the literature, but we wanted to understand. So what we did was we have a genome, like the regular one I had, and now we have a trait that is cooperation or defection that you are learning, I think, right now, right? And we wanted to understand if we could form species based only on this trait. This is a trait, right? As you know, we have many different games, and one of these games is the prisoner's dilemma. And what did the prisoner's dilemma predict for the mean field theory? Is that defection will evolve. Doesn't matter if the cooperation gains more in the end, defection will evolve, right? You already learned about that. And Novak, I think you have the book already. Novak has already shown us in the past that the spatial configuration is something important, right? The spatial configuration can make clusters in the environment where cooperation can evolve and maintain. And we wanted to understand if not the space, but the speciation could be the ingredient that could make cooperation remain in a given population. And okay, we analyzed it. We wanted to understand if these things with and without the game were similar. So we did exactly the same thing. We saw the species were forming, and we saw the cooperation frequency could be higher, could be lower, depending on the size of the payoff. And here's just variation of the payoff value. But more than that, what we found is that the balancing and the acceleration of the phylogenetic trees that were formed was different. Without the game and with the game, there was a big difference here that we could notice. So the natural selection is actually playing a role to form these phylogenetic trees. And this was something that we could show with this work. This is still ongoing, and Luis is still analyzing some data, but something that we are interested, and that's why we did the previous work. We wanted to understand how extinction, fusion, and speciation reversal, this is wrong, plus the game theory could show us some things. For instance, if cooperator species vanish more frequently, if hybridization mixes species of different strategies. So to know that, we needed the previous work. And this is something a student of mine is doing right now. So as things that we are working on and I'm willing to understand is exactly how natural selection combined with extinction and hybridizations, how we understand the play of natural selection on extinctions and hybridizations, how cooperation evolve under different games, not only prisoner's dilemma, and the signals left by different types of mating, like assertative, disassertative, and neutral. This is another ingredient that, not with the game, but just what is the assertative play and the evolution of species. And also understand coevolution, so simulating different initial populations, like plants and animals, and how we can see combining. And, oh, sorry, this. And that's it. If you have questions, thank you, gracias. You didn't sleep? Are you still awake?
I'm sorry. Sorry, Ricardo. I want to ask about the last part, about the diversification work. And it's really a question because I think I got lost at some point. So
Me too, every day.
so that model, the genome, you are considering individuals to be haploid or diploid?
To be what?
Haploid or diploid? One or two?
Haploid.
Haploid. And when they reproduce, what happens exactly?
It's like this. Is it work? Yeah. Sorry, I'm yelling. So let's say you have the, I don't want to say male and female because we don't have sex here, but we have parents. I'll call P1 and P2. And what happens is that with the probability of 50%, this will form the offspring here. The allele will be inherited by the P1 and 50% from the P2.
Okay.
It's just like that. It's a way to simulate the crossing over.
Okay, that's what I was
We
thinking.
also simulated crossing over, like the typical crossing over, like you do a given point here, you choose the point where you're splitting. This is just faster than this.
Okay.
It's about the same thing.
Yet about this.
That?
Yes.
Okay.
Is that something which reproduces in such a manner? Because when I think of crossing over, I think in the same individual.
Yeah. That's true. Here we are simulating a crossing over between different individuals. It's just a way to actually it's just a way to say the genes are independent, right? To be passed to the next generation. Oh.
I have a question as well. Okay. Yeah, about the role of space in your model.
About which one?
The role of space in this last model as well.
Okay.
Okay. Is movement something that makes such? I think there are two limits that I can imagine this model existing, which to me seem in some sense more tractable. At least they look closer to, let's say, some kind of model that could be treated analytically. One would be the case where movement is very zygotic, and then there is a kind of well-mixed population. And then there's the limit where these things are really just interacting with very close neighbors. They basically don't move in the relevant scales for evolution. And then it's almost like some kind of network interaction, right? I think this model seems to allow for all the intermediate things here. But does it ever play like, does this particular way of implementing space ever play like a very important role in the dynamics?
So about the movement, we can say that in the sympathetic scenario, where we have like, let me draw here. It's easy drawing. So we have our lattice here and an individual here. If the radius to look for the mating partner is small, it moves less, right? If the mating area is large, then it's like moving around and finding a partner, right? So it's kind of similar of the movement, but not exactly. But I don't know how to translate one in the other.
Maybe I didn't understand. There's no movement here at the level of these individuals. They are just, they stay in space. There's this radius.
Yeah.
They interact probabilistically within this radius, and then you throw a new individual in.
Yeah.
Okay.
In the same place of the focal parent or around.
Ah, okay. So the spatial structure is already a kind of network, but it's kind of represented in a
So
more
when
spatial.
when you have the mating area is very large, you don't find this structure that you're saying.
Mm-hmm.
It's very spread. So you have individuals of different species everywhere. Is
Ah,
this?
okay.
So
Yeah.
when you have like those, when the mating area is very small, you see those groups of same color like in this organized in this space, right? It's a product, it's a byproduct of the mating area that they are using.
Okay.
So when then you increase the mating area, the individuals of different species will be everywhere. And like, I don't know if it's random I cannot prove it's random, but if you look and say, oh, it's random. It's how we choose the partner.
I was going to ask you something similar to what Benjamin asked about movement, because I was thinking about resources. In nature, there are places that have more resources than others. So maybe if you put some gradient of preference of movements, you could have something interesting. And I was going to ask this, but now I know that you put this area. Okay. So, but I have other question that is related to the lizard experiment, or lizard, the three lizards, the blue.
Yeah.
Yes. You could see that you change the configuration of the survivance of the coexistence or not, but you have data about this in real life, like
Yeah, there is data. Sinervu and his group have a lot of data in different areas in the US of the frequency of the different kinds of males.
and you are able to compare with the results, and the other result was better?
So they compared in their work. We didn't compare in ours. We just wanted to understand what happens when we join these two kinds of informations, that they have different behaviors, and these behaviors is defined genetically. So we wanted to join and understand how joining them would mean in theory. But we didn't compare with the data from the field. And also, you didn't ask, but I'm giving you a piece of information that I didn't give. We have females, right? They are diploid individuals. So something is coming from the female, I hope. If everything works like we know, something's coming from the female. And the females also have different strategies that are related with the OBI system, with the alleles that they have. I don't remember right now how this is translated, but one of them is what we call in biology R strategist, and the other one is K strategist. And when we say this about reproductions, when we say, we mean that some of them have many offsprings, but they don't take a lot of care about them. And some of them have very few individuals, very few offsprings that they care a lot. They give a lot of food or something like that. And so the females also play a role, and to understand the frequency of individuals in nature, you need to understand females as well. And unfortunately, the student that was working on this more is already doing something else outside of academia, so we stopped working on this, but it's something that is a side project. It's in my desk. At some point, I will retake it.
My question is more related with that kind of matrices, payoff matrices. And in ecology, how can you assign the values in the matrices?
Oh, how you explain the payoffs, for instance?
Yes, I mean, what characteristics or how do you measure the things to assign the values?
I like this kind of question because I also don't go to the field, but I want to understand that as well. But it's related here, it's related to the number of springs a given male leaves compared to another male. So the orange one, let's say it leaves one offspring when it's competing with another orange one. And if it's competing with blue one, because it has more females, it leaves more offsprings compared to the male one. And because the yellow one sneaks some females of these guys, it decreases the offsprings that it would leave in competition with another one. So this is very theoretical, I agree, but and what they usually look in nature is to the result, is to the frequency of different strategies, not exactly to these payoffs. And then you can maybe adjust this. Once you understand the system, you can adjust the values of these payoffs to get the frequencies that you observe, for instance. But they know very well the system. They are amazing. And they know like about everything what these lizards are doing. So it's amazing. I'm kind of sure that they know the number of springs each male leaves. Like in competition scenarios, I think some of these, some of the empiric experiments were done in the field as well. Who is?
You said that it doesn't, it wasn't an assortative mating choice, but if you define a small distance between the genetic correlation between the partner and the focal, it wasn't be an assortative mating choice?
That's
because they are similar?
That's a question that I get in many of the talks, and it's hard to confront biologists. But when we say the mating is assortative, is that we have a preference. They will reproduce more likely with those that are similar, or just reproduce with those that are very, very, very similar. This is assortative mating. And some people say, oh, if you have this G value, and then if it's smaller than G, it's not assortative. It's a level. So we are analyzing different scenarios of assortativity. But we just wanted that individuals to reproduce among their co-specifics. Because we had to define species somehow, and that's the way we define species. So we are now analyzing different levels of assortativity to understand it better. And it's very impressive, the power of assortativity in forming species. Like if you have a very huge genome and just a very, very small portion where you choose based on that portion, the species that are formed based on that small portion or based on the whole genome is the same. So there is somehow a hitchhiking effect, or this small part, the assortative part, is carrying the rest of the genome. So it's very powerful. So we are analyzing these different assortative scenarios, but I like that question. That's why we developed this work, because many people make this question. Yes.
Hi, Flavia.
Hi,
my question is more towards the beginning of the presentation when you talk about rate-induced transitions and the pollinators. Yeah, I got confused and kind of lost, especially in the next slide, because you talk about like
there's a lot of information here.
Yes, and the no, but what really like scrambled my mind when you said about the reweighting, like redistributing the weight. When you distribute the weight, the relative persistence drops.
Yes. That's counterintuitive.
Yeah.
So we were, I will explain again just to move to that part, okay? It's just to meet myself as So, oops, sorry. So here we have the rate of change of the driver of decline. So the parameter is changing in a given rate. Here is fast, and fast is like this. Here is slow, and slow is like this. And they reach a maximum that can be low, medium, or high, but all of them, we would not observe a collapse in the previous scenario, okay? So what we observed here is that in this scenario where we maintain the network as it is, we don't change anything, if the rate of change is high, when we reach the closer to the maximum that we could, we have extinctions, we have collapse. And if this rate of change is small, it doesn't happen. For the other scenarios, we don't see any change. It remains as it was predicted. But when the species adapt and they redistribute the weight they have with their partners, as like, for instance, as extinctions start to happen, they redistribute their interactions. What happens is that even in the medium case, we kind of shift to a different scenario. And for the previous one, for the orange, that is the worst, close to the worst, it can happen in a smaller rate. And that's, yeah, so maybe species are not adapting like that. They are not like redistributing the weights of their interactions. Maybe it's what is happening, but maybe it's what is happening. And if it's what is happening, it's worse, right? We need to consider that. And maybe species don't know the future. They don't know what is going to happen. They are adapting to the present. And what is going to happen in the future, because they adapt, is also affected by adaptation. So, yeah, that's...
So one final question.
Okay, I have two questions.
So make one and then the other, otherwise I get confused.
No, just make the one. Back in the simulations, I was
Which
curious
one?
About the non-continuous one, when you added the barrier.
Sorry.
The simulation of the individuals when you added barriers.
Speciations,
yeah. I just wondered, did you start the simulation with the barrier between the individuals?
If you start what?
The simulation with like the barrier between individuals, you did, right?
Start a simulation, I don't understand.
With a barrier, with a barreira.
A barrier, oh yeah.
You started the simulation with it. Did you try to add the barrier like in the middle of simulation?
No, some people did that already.
Oh, no, what happens?
But they included so many ingredients. I hate that work, but I like it because they did something that I don't want to do. But they included competition, they included barriers, they included something else. If I remember well, it was, the first author is Pigot, but I need to remember that. And I don't remember the result, but I can get you the reference.
And the second one was just like the reference for the reticulated evolution. I think I missed it. Could you like pass to the slide?
Mine or?
Yeah, no, yours.
Oh, it was published like 2 days ago. But it took like 3 years to do that. No, 2, maybe 2, just the pandemic. I didn't put it. It's not here. It's Botelho. Marquitti and De Aguiar. 2022, Journal of Physics something. It was in a physics journal. But I can send you. It's open, I guess. If it's not, I'm the owner. And that's it. Thank you, guys.
Thank you, Flavia.
Thank you.
And we'll see you tomorrow.